Second note set is on a closely related molecule to DNA called RNA. So this is page 2 of the notes. RNA stands for ribonucleic acid. Okay, it's also called messenger RNA, and the little m is the abbreviation for messenger. So what does a messenger do? Well, they carry a message from point A to point B. So you have a mailman or mailwoman will carry the mail that gets delivered for you to the post office, and they bring it to your house. So that's a messenger. If you put something in your mailbox to be mailed, they will take that mail back to the post office and it gets mailed. So that's a messenger. And messenger RNA looks like this. So this is your structure of RNA or messenger RNA, and this is the structure we talked about in the first notes, the structure of DNA. Visually, how is the DNA different from the RNA? Well, one, DNA has two letters facing each other. So we call RNA single-stranded. There's one strand. This green and orange is the backbone, which we talked about yesterday. So that's the backbone. And then you have your nitrogen bases. Well, DNA has two strands. Here's one strand. So you have your backbone, your phosphate and your sugar alternating. And then you have your bases. And then you have another strand. Only this strand is kind of turned upside down a little bit, and the letters face each other. And then we talked about yesterday, there's hydrogen bonds here. RNA does not have hydrogen bonds. RNA is only half the width of DNA, so it is single-stranded, whereas DNA is considered double-stranded. And what do you notice about the letters? Okay, here's a U here. There's no U in DNA. So those are just visually some differences between the two. You notice the green circles are the same, but this is different. The DNA, the deoxyribose, is colored orange. The ribose here is colored yellow. So why is that? Well, yesterday we learned deoxyribose is the name of the sugar in DNA. This is a picture of it. This is the sugar ribose. You notice that if you compare in the exact same location, everything about the deoxyribose sugar is identical to the ribose sugar with the exception of that red O. That is oxygen. So deoxyribose, it's named that way because it is the same sugar ribose, but D means missing or without. Oxy is oxygen. So deoxyribose is the sugar ribose without the oxygen. And see, it's missing right there. So deoxyribonucleic acid, the ribose sugar in DNA is missing the oxygen that the ribonucleic acid, the sugar ribose in RNA, has it. Okay, so let's take a look. The job of mRNA, it takes the directions for one gene and transports it to a ribosome in the cytoplasm. So it's a messenger. It takes the DNA gene, the code, and it translates it, it transfers it out to the cytoplasm where it lands on a ribosome. The subunit of RNA is also called a nucleotide. So it's the same as it was yesterday. It is still made up of a phosphate. There's the blue phosphate. It's still made up of a nitrogen base, though we know there's a new one now. But the sugar is ribose. So this is a ribose sugar, not a deoxyribose sugar. But they're both nucleic acids. So the building block, the subunit, is identical. It's just the sugar's different, and then there's a difference in the base. So here is our nucleotide, and there's 4 different nucleotides. So which base is new? Okay, the uracil is the new base. So A still stands for adenine, C still stands for cytosine, G stands for guanine, U stands for uracil. And they're using the first letter of the word again as its abbreviation. So for RNA, this question mark here is going to be only one of these 4 letters. Notice the T is not there. There is no T or thymine nitrogen base in RNA. Now, transcription. Transcription is when DNA is used to make mRNA. And this occurs when proteins need to be made in the cytoplasm. So somehow in the nucleus, a gene is activated. And we talked about in differentiation, when we're growing up in the womb, is that these little baby stem cells become differentiated adult cells. And so the genes that a cell no longer needs are permanently shut down. Transcription can only occur on genes that are still in the on position. So if you're a stomach cell, your cell can make stomach acid because that's its job. Okay, if you're an eyeball cell, cells in your eyeballs cannot make stomach acid. That would be a bad thing. So in this transcription, which is an unusual word, you're taking the DNA code and you're somehow making mRNA from it. All words in RNA are called codons and are made up of 3 letters or 3 nucleotides, just as DNA. So this diagram here shows this gray strip is that alternating sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. So this picture just turned that into a ribbon to make it less confusing. And then these are the 3 nitrogen bases. And a codon is a word in DNA language. It is also a word in RNA language because remember, they both belong to the same macromolecule group, nucleic acid. So they have the same building block, the same purpose. All right, transcription. This is how it works. So here is, and this is in your notes, a piece of DNA reads. So this is the DNA, and we know it's DNA because there's a T here. So TAGCAT-TCC. That's just a little strand of DNA. Transcribing it into mRNA. Okay, well, this is the way it works. If there's a T in DNA, it bonds with an A in RNA. So there we go. There's our A. So T in DNA binds with A in RNA. An A in DNA cannot bind with a T because RNA doesn't have one. So for DNA transcribing into RNA, A binds with U. Okay, we can't put a T there because look in this RNA line, there's no T. The G and the C's are always the same. So this is the faster part. So you know G bonds with a C, C bonds with a G. Okay, now what bonds with that A? Look over at your chart. U bonds with A. Those 2 T's. If DNA reads T, there it is. So that should be 2 A's. And then the C bonds with the 2 G's. Okay, so this is how your transcription. Now, here's another grammar issue. The descriptor or the adverb is called transcription with a P. The verb is called transcribe with a B. And I know that's a bear, and I'm sorry, that's English. Okay, that's the English teacher. I have nothing to do with that. So transcribe is telling you do it now. Transcription is like an adverb. So it's a description of the process. So they mean the same thing. One has the B and one has the P. All right, so let's practice. Okay, so if we come here, transcribe to mRNA. Looking at those notes, what letter bonds with A? U bonds with A. G is easy. C bonds with G. T, there is an A in RNA, so that's going to be an A. The A Is there a T in DNA? Nope. So that gets a U. Okay, so if I transcribe the rest of this, now a way to look at this is, and this is the way some students do it, they transcribe it. These used to be T's when we were doing DNA. So when you're transcribing mRNA, look back at your answer. If you see a T in there, take the T out and put a U in it. Okay, so this is going to be, that's going to be your answer, transcribe. So try this one. Oops. What bonds with an A now? Do I put a T there? Oh, G. C bonds with G. That one's the same. There is an A in RNA. Here we go again with there's A's. So if this were DNA, that would be T, but there are no T's. So that's going to be our U and then C. Okay. This one you're going to do in the video, or in the Play Pause It video. Okay, so it's just practice, and you're going to get that today. Now, the last thing I'm going to leave you with is a video that, we're going to actually watch right now. Okay, and it's less than 2 minutes.
What you are about to see is DNA's most extraordinary secret: how a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U.
Okay, so this is an enzyme. This is your double-stranded DNA molecule. This enzyme unwinds the DNA, uses one side of the DNA, and transcribes it into mRNA. Everywhere there's an A, there's a U in this yellow RNA. So if there's an A in DNA, there's a U in RNA. Otherwise, all the letters are the same. Okay. Oops.
You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
Okay, so that is it for this section of notes. You're going to get some practice. So good luck.